To date, you can measure mutations, single mutations, that is, in several solid tumors, and they also steer the treatment of patients. The most popular or most known example is, for instance, KRAS mutations in colorectal cancer, which would tell you that a certain drug, cetuximab, would work or wouldn't work. Now, there are more than one mutations in any tumor, and with the NGS, that is next-generation sequencing, you have the chance to really take into account all of the possible mutations which are relevant. Relevant in this context means that they are druggable, that is, there are drugs which can be directed against this particular change, which is then called a biomarker. Next-generation sequencing means that you're doing a high-throughput, highly parallel sequencing. And this has been extremely expensive, and the prices are coming down, which is a very nice thing. This kind of sequencing these days can be called a commodity. You know, everybody can do that. So the real question is about, the question is garbage in, garbage out. So you really have to make sure that good material, good tissue, good DNA is going into this kind of analysis, which is still rather expensive, and then take these data and put them into an evidence-based software tool, munch them, crunch them, and in the end come up with a report which would tell you, well, these drugs are evidence-based, recommended, plug one of those, and then there are other drugs maybe which are, you know, evidence-based, shown to be non-effective, and in the end, drugs where you do know, because of the inborn errors of metabolism, as they are called, that there is increased toxicity to be expected. If you look at, say, a typical sample, you have some, say, 30,000 mutations maybe. Not all of them are relevant, because we all carry certain abnormalities, I should say, which don't have to do anything with disease, in this particular case, cancer. So the idea really is to take all of these data together and put it in a black box, and in the end there will be a comprehensive report which a physician, in this case an oncologist, could use. And that would mean that you take into account all of the mutations which make sense, say, in a signal transduction, so that you can really determine what would be a recommended drug a patient could use, or you as a physician rather. It's not only a revolution, I might say it's a paradigm shift, because the way you have looked at it and the way industry has conducted studies is they have a drug which is in these days a so-called targeted therapy directed, say, against a particular biomarker, a particular mutation. Now, this may occur, say, in colorectal cancer, but you find it also, say, in pancreatic cancer. So the way you look at a tumor will not only be from the site where it originates and, you know, the histologically makeup, but it will be made up by the molecular profile you find. And you can have a molecular profile, say, in ovarian cancer, which is basically similar to one which you would find in the colorectum. So as a consequence, you should treat those patients differently. And that's what is rather new about it, and which is a big kind of challenge to everybody. The physicians have to learn to use drugs they normally don't use. The payers and the insurance companies have to learn that they should, you know, pay these kind of things. But overall, it's very, very difficult to create and to produce evidence for these kind of studies, because clearly you cannot take one company funding one drug and then pursuing a study in one tumor entity. So there are a lot of, you know, challenges these days in oncology. We are conducting a study where the health economy is an integral part of our study. That is to say, these tools where you now look for all the biomarkers, genetic changes, not only look for markers which would allow you to take a certain drug, it will also allow you to avoid drugs which will most likely not work in a given patient. It will also allow you to determine the inborn changes which would make that you or me would react with higher toxicity against a given drug. So the upside of this personalized cancer medicine is also that you would avoid additional costs. So We don't know yet, I have to say, but it might very well be that in the end, if you take all these elements into account, into the equation, that it will not only be better for the patient in terms of survival and even quality of life, but it might also be cost effective.